Hello everyone, this is Chippy and from the Department of Pharmacy Practice 9 for Guwahati, and I'm here to present my research title Regulatory Role of Placental MicroRNA, specifically MIR26B5P and MIR4985P in hypertensive disorders of pregnancy. While discussing about hypertensive disorders of pregnancy, the major organ to be focused on is the placenta, as this is a placenta mediated complication. As you might know, placenta is an organ which is developed inside a woman's body during her pregnancy, and it helps with the gas and nutrient exchange between the baby and the mother. So any dysfunction associated with placenta can cause severe complications. Now I would like to introduce you to my research area, that is placental microRNAs. These microRNAs are short RNA sequences which have the capacity to regulate gene expression. So we can call these microRNAs as a kind of editors of gene expression. It decides whether the gene has to be upregulated or downregulated in a human body. Our study started with an initial transcriptomic analysis in which all the placental microRNAs were quantified of both the groups, control and disease group. And from the data analysis, it was found that two of these microRNAs were found to be significantly downregulated in the disease group. We selected these two microRNAs and further validated in an extended cohort of samples. We have collected clinical data and placenta samples from this cohort of samples. MicroRNA was isolated from their placenta samples. It was converted into cDNA, and the differential expression of these two particular miRNAs were measured using qPCR. Further, we have correlated this data with the clinical parameters, and also receiver operating characteristic curve analysis was conducted to identify the discriminatory ability of these microRNAs in predicting the disease. Also, using bioinformatic analysis, we identified the molecular mechanism through which these microRNAs act. Our study results showed that our both microRNAs of interest is found to be decreased in the gestational hypertension group when compared to control, and as the disease progresses to preeclampsia, it is more decreased than gestational hypertension. Also, clinical correlation analysis showed that the clinical parameters, that is blood pressure and total protein, was found to be negatively correlated with the microRNA expression for both our microRNAs. Also, ROC curve analysis showed that there exists a high discriminatory ability for both the microRNAs in between the control and the preeclampsia group. Also, the KEGG enrichment pathway analysis showed that for 26B5P, the major molecular mechanism is MAPK kinase pathway, and for 498-5P, it is WNT signaling pathway. So, in conclusion, our study shows that these microRNAs may help predict the disease, and also it shows the molecular mechanism through which it might act. That's all. Thank you.